Hello everyone and welcome to another video. It's been a long time since I posted a long and juicy topic for you to get stuck into, and there are many reasons for that. But today we'll be discussing haemoglobinopathies. We will look to understand the production of hemoglobin and its normal structure. We'll also touch a little bit on where this can go wrong and why that's significant. We'll dive into more detail in subsequent videos. I'll be joined today by Daniel Monteiro, who is a principal biomedical scientist in special hematology red cell laboratory at Synovist, based at Guy's and St Thomas's NHS Trust in London. Daniel's main interests and expertise are in the screening and diagnosis of haemoglobinopathies. His experiences encompass both DNA and protein-based techniques. He did his initial training in Portugal and subsequently moved to the UK and did his MSc in biomedical science at the University of Westminster. Besides the projects he actively participates in relating to haemoglobinopathies and haemoglobin studies, he is involved in the training of all levels of scientific and medical staff as the training lead for the lab. I can attest to his training prowess, as when I was doing my IBMS specialist portfolio in haematology and blood transfusion, I too had the opportunity to be trained by him during my rotation in the special haematology laboratory. In addition to this, Daniel also lectures at the NHS Sickle and Thalassemia Screening Programme, Training Days, Annual Sickle Cell and Thalassemia Conference, and also has presented at the IBMS Congress for multiple years. So Daniel, my first question to you, what exactly is a haemoglobinopathy?
Haemoglobinopathy, depending on what literature you read, has various definitions, but in essence itself, it's everything that could affect the synthesis, production, or function of hemoglobin itself.
Okay, so why is hemoglobin important?
Well, hemoglobin is not just essential for life, because his main properties or functionality inside the human body is to transport oxygen from the lungs to the periphery, or every tissue is dependent of that little molecule out of hundreds of it, just to be able to provide oxygen to the tissues, which all of them require. So if something is wrong with it, you will have limited capacity to provide that, and you're going to have physiological effects and diseases coming out of it.
So without hemoglobin, basically, your tissues can't do what they need to do. They can't respire, they can't make energy, etc., to do their functions. Okay, so you mentioned a little bit about the abnormalities that can occur in hemoglobin formation and production. Do you want to tell me a little bit about those and tell me a little bit about hemoglobin? What's the normal production of hemoglobin, and how can that be affected in a hemoglobinopathy?
Absolutely. So there's a couple of slides we're going through, and we're going to look much more how really taking step to the origins, the basics, because when you're talking about hemoglobin, it's always good to remember what is there to actually first produce it, because many of the abnormalities, many of the hemoglobinopathies come from where hemoglobin is produced. So we'll start right there from our first slide, because we need to know how hemoglobin is synthesized and how it's formed. So from here, you can see that the hemoglobin synthesis is done, obviously, in genes. Particularly for hemoglobin synthesis, and especially for the main hemoglobins in a healthy patient, which we're going also to talk in the next slides, there is specific genes involved in its production. But before the hemoglobin is actually produced, you kind of like need to produce the puzzle pieces required to build it. So to build the main hemoglobin fraction, and that the majority of we have, you need alpha chains and beta chains. Those are quite easy to remember where they are produced in the genes, because it's also called alpha genes and beta genes. So it makes it much more easy to understand. So on chromosome 16, you have the genes involved in the production of alpha chains, the alpha genes. And on chromosome 11, you have the genes involved in the production of beta chains, so various amino acids that produce polypeptides. And you also have the delta genes which produce delta chains, which we're going to discuss what actually they're involved with. But any gene needs to be transcribed first to mRNA and then translated into polypeptides, amino acids in essence. So now that you have the puzzle pieces, or the Lego pieces if you want, you really just need to put what you have available together to produce the hemoglobins that come out of it. So putting alpha globin chains with gamma globin chains, and you produce hemoglobin F, F for fetal. So again, it will make it more easy to understand because it's the main hemoglobin presence when the babies are born, and also in the later stage of gestation as well. If you put together alpha globin chains with beta globin chains, you produce hemoglobin A for adults. Easier as well to understand because it's going to be the main component of the main hemoglobin present in adulthood. And then you just see what's remaining. You have the alpha chains, and you put the together with delta globin chains, and you produce the hemoglobin A2. Now, it really depends when you're doing analysis and when you look in the hemoglobins that you have present in certain amount of time. It needs to be in context of how old is the patient that you're looking at, on which point, which age we are talking about. Because as I said, hemoglobin F is the main component present in when babies are born. So you can see here from the point of birth, it's going to be pretty much prevalent, but it's going to rapidly decrease, and it's going to produce the hemoglobin A for adults, and also a small fraction of hemoglobin A2 throughout the time. So, but you reach, should we say, an adult phenotype. So you're going to have all hemoglobins, normal hemoglobins present pretty quick, because around 1 year of age, just 1 year of age, you're going to have all hemoglobins that you're supposed to have for the rest of your life. So this is where they are produced, how they are formed. So how much are we actually talking about? Because haemoglobinopathies, when is very, very much summarized in the table, in adults, you expect to have very small fractions of what you had when you were born. So hemoglobin F. Hemoglobin A is going to be, again, the main hemoglobin that is going to be present, completely functional. It's going to do all the functions which are essential for life that we already discussed. Hemoglobin A2 is really small compared to the other ones. So it could even be argued it doesn't really have a function, a physiological function in the body. However, it's quite important for diagnostic purposes. So especially in the laboratory, and we are diagnosing carriers of certain haemoglobinopathies or even diseases, hemoglobin A2 is quite important. Now, haemoglobinopathies itself can be quite complex in a way, because there's a lot of interactions can happen, but the core is always the same. Because if you have limited amount of genes involved, you're also going to limit the possibilities of diseases or what can happen. But you can segment haemoglobinopathies and the main two categories, which are first thalassemia. Sounds a very complicated name, but in essence just means that out of the genes that we just looked at, some of them are not producing, shall we say, the normal quantity that we're expecting to produce. So it's a decreased or reduced rate of synthesis of the certain polyglobin chains that we discussed. And depending on where the mutation or the causative factor is, could be called beta thalassemia or alpha thalassemia, depending if it's on the beta genes or the alpha genes. So hemoglobin variants, and that is one of the main ones we're going to discuss and much more prevalent in our studies, is actually something new is created. There is a mutation sitting somewhere in the genes that instead of producing a certain amino acid, is producing a different amino acid, which then when the polypeptide and the hemoglobin molecule is formed itself, it's different. So if it's different, it's called a variant. And many of them are quite important for us to diagnose because they're going to have some clinical impact. They're going to have some capacity that is going to have consequences physiologically. There's other components, and I left it a bit out of the main components, which are, for example, when you start to produce normal hemoglobins, but in an unusual amount that you would not expect to produce, for example, in adulthood. I said that you expect to find very fractions of hemoglobin F or none at all. But sometimes you have mutations that will actually produce a lot of it, even in adulthood. It doesn't necessarily mean you're going to have a disease or having consequences physiologically. It's much more important on the stage when you're possibly looking at having a family, because that all of this is transmitted or genetically inherited. So it's things we need to look into, not only what the patient is looking now or how he's faring, it's much more about what he can transmit as well to future offsprings as well.
Quick question for you, Daniel here.
Yes.
So you just mentioned that it's all genetics. Does that mean you can't acquire haemoglobinopathies throughout life? You're born with it, basically, as Lady Gaga song goes.
In essence, yes. All of them are genetic based, so it's not something that you can develop. Even if you're exposed to a lot of mutagens, radiation, etc. But haemoglobinopathies, in its broad sense, it's just genetically inherited. So it's all good to know which genes are involved in the production of hemoglobin. We know the function of the hemoglobin, and we know what, in a theoretical sense, what can be wrong in its production of creation of an hemoglobin variant, something abnormal. But what's the purpose of all of this? Is it actually clinically significant? Does it actually make any difference of the people that actually have hemoglobin variants? So we're going to see a short video that is much more looking in one of the more prevalent diseases out there, and you might have heard it in social media, news, and many places as well, called sickle cell anemia, sickle cell disease. Yet again, it's good to know that those terms are available out there, but actually, what does that mean? If a patient has sickle cell disease, what does it actually happen in the patient itself? So is everything going to happen in your vessels, so where hemoglobin is present? Sickle cell is a very simple mutation, which we're going to see, but what actually makes, instead of a nice round shaped cell that is very malleable and very flexible to go through capillaries, which are even smaller than the red cell itself, they become sickles, like the shape that we see. So they become very rigid and also sticky. So you can imagine that if it's sticky, it's going to basically block your circulation and having consequences. But all of that because only one nucleotide after the gene, only one has been changed. That change makes that amino acid that is produced out of that exon is going to be different. So it's going to produce a different hemoglobin, hemoglobin S. That hemoglobin S is very sticky itself by itself. So it's going to create quite long segments of hemoglobins inside the red cell, pushing and creating edges on the cell itself, and hence became so rigid. So the problem with it physiologically is when you actually get into the circulation and as sickle cells are formed, they will not be able to go through those capillaries, that circulation. And it happens quite frequently where microcirculation is quite frequent. The brain, the eyes, the liver, pancreas, bone, all of those are quite dependent on a very small, tiny capillaries or blood vessels. If all of them, or a good amount of them, are blocked because of these sickle cells, you don't have not only oxygen supply, but blood supply. So those tissues itself are not going to be performing or even survive for a long time if a lot of these cells are produced. So it can be life-threatening, and hence hospital admissions are not that uncommon. So it needs to be really well monitored, but that is the significance of one amino acid change, one hemoglobin variant. And I must say, there is more than 1,360 hemoglobin variants described. We're just talking about the hemoglobin S, which is not only one of the most prevalent and clinical significant, there's a lot of combinations that can happen.
So I guess my question here will be, given that you've mentioned there's more than 1,300 different hemoglobin variants that you can get, how do you diagnose or how do you screen for them in a hospital setting?
With luck and a good amount of experience. So the luck part is joking around, but it is something that needs to be looked at in a quite broad sense. it is not difficult to separate hemoglobins out. It doesn't really matter how many hemoglobin variants you have, but the first thing you need to have is a way to separate them out. Even just normal hemoglobins, I've discussed the hemoglobin F, A, and A2. There's 3 of them. But when you take a blood sample and it's sent to laboratories, one tube, one bit of blood inside the tube, how are you going to focus on not only that little bit of sample, but there's lots of things inside that sample, lots of cells, lots of molecules. How are you going to focus only in one inside the red cell on top of that? So various technologies have been appearing throughout the years, and the most common ones is, for example, HPLC, which stands for high performance liquid chromatography. There's various others like capillary electrophoresis, but instead of going through in the principle itself, it has the capacity, and that obviously there's various companies that create different technologies that I'll be able to not only separate out all the hemoglobins, but also focus only on them, and everything else is not measured or quantified. So on the picture itself, on the slides, there is it's automated essentially because it needs to be quite reliable and reproducible. So that these days only acquired by analyzers. So it needs to be automated. And there's a couple of pictures of analyzers that are used much more frequently, especially in the UK. So throughout the world as well. So that is what is called a screening test. You're separating out the hemoglobins as much as you can, focusing, and it's absolutely mandatory for you to find the ones that are clinical significant, such as S. So after separating them out, you're going to quantify as well. Everything is done by the analyzer, but it's not as much of analyzing a sample, is how you're going to interpret the data that is presented in front of you. So screening tests are there to be rapid or moderately rapid. For example, we use an analyzer that each sample takes 6 minutes to separate all hemoglobins. In context, for example, you do a full blood count, which takes a matter of seconds or sometimes. So it's a more prolonged analysis. You need to separate hemoglobins. And from that point, you're going to see what's abnormal, if there is any abnormality. So that's your screening. You imagine that you find something abnormal, such, for instance, hemoglobin S. The next stage, which you need to do, is use a confirmatory test. It's not just good enough for us to be looking at there is an hemoglobin variant detected by one analyzer or one technique. You need then to use a second technique, which is different than your first, different principles, so you'll be able to confirm it. That is absolutely necessary. Hence the fact, well, essentially because if there is more than 1,300 hemoglobin variants described, they can be anywhere, even on the same places where other hemoglobin variants appear. So you need to have a second test just to make sure what you've seen on the first test that you've done is actually correct. And that is a various tests that you can do, semi-automated, manual, more fancy or less fancy. So it could be, and that's some lists. Gel electrophoresis is quite much common, especially in university settings, for instance. We do actually do gel electrophoresis. That could be one of the techniques, but you could go a bit more in depth and do DNA analysis. You could test the genome itself to see what the variant is. There's various techniques, and each one has its advantages and disadvantages as anything. So I hope this introduction was good, but essentially that is the importance of doing. And there is a national run scheme or system, which is the screening program, which is looking at this. Not only when babies are born, the heel prick, the sample that is taken out of newborns, actually is looking into haemoglobinopathies as well as any other many other diseases, but haemoglobinopathies is one that is also looking at. But also when families or couples want to have babies, there's also the antenatal screening program. So one of the tests that is done when a lady is pregnant is looking into haemoglobinopathies as well, just because what can she pass to the baby needs to be analyzed and see, because what we want to minimize is that a lot of babies are born with diseases, well, at least the parents will be informed about it as well.
In the next few videos, we'll look at some specific haemoglobinopathies in more detail, understand a bit more about some of the screening techniques Daniel mentioned, and also how the results can be interpreted. Daniel and fellow biomedical scientist Sarah also have a YouTube channel called Haemopaedia, which focuses specifically on haemoglobinopathies. It's a work in progress, but if you'd like to visit, then I've left a link in the description below. Thank you for watching the video. If you found it enjoyable and informative, please do give a thumbs up, and if you loved it, hit the subscribe button below.